When developing peptides for pharmaceutical use, the science isn't just in the design, it's in the verification. Peptide testing is what ensures that the molecule you intended to create is the same one you're actually working with, consistently and reliably. In this session, we'll walk through the key considerations you need to understand before outsourcing peptide testing, so you can make strategic decisions that protect your development timeline, your data integrity, and your chances of regulatory success. Peptides are used across biologics, vaccines, targeted drug delivery, and even diagnostic technologies. But peptides are sensitive. Small changes in manufacturing conditions, how they're stored, or how they're purified, can dramatically change their behavior. This can lead to shifts in activity, stability, safety, and ultimately, regulatory outcome. Testing is what confirms that the peptide you designed on paper is actually the peptide that enters clinical development. It's about confidence, reproducibility, and risk control. Before you outsource testing, there are 3 questions to get absolutely clear on. First, what exactly do you need to characterize? That may include sequence confirmation, purity testing, structural analysis, or biological activity. Second, what development stage are you preparing for research? Preclinical, clinical, or commercial manufacturing? Your testing depth will differ depending on where you are. And third, are you testing for purity, identity, stability, functional activity, or all of the above? Getting clarity here prevents wasted money, repeated testing, and lost time. Peptide testing relies on specialized analytical techniques. LC-MS or LC-MS/MS confirms your sequence and detects impurities. HPLC or UPLC measures purity, which is critical for regulatory documentation. MALDI-TOF is used early on for fast mass confirmation. Circular dichroism helps evaluate folding and secondary structure when the shape matters. And stability studies, guided by ICH standards, show whether your peptide will remain functional over time and under real conditions. A capable testing lab should be experienced in all of these, not just 1 or 2. When someone says a peptide is 95% pure, that sounds great. But the real question is, 95% of what? Peptides often contain truncated fragments, deamidated species, oxidized variants, or aggregates, and each of these can influence behavior and safety. Regulators don't just care about the purity percentage. They care about the identity and quantity of impurities, how well they're controlled, and whether you can track these impurity profiles batch after batch. So resolution and impurity analysis matter just as much as the purity number itself. Even a perfectly pure peptide can fail later if it's unstable. Peptides degrade through hydrolysis, oxidation, aggregation, and racemization. If this isn't detected early, the result can be lost potency, safety concerns, or complete product failure during scale-up. That's why stability studies need to mimic real-world conditions. Temperature fluctuations, formulation environment, exposure to light, and freeze-thaw cycles. A strong testing partner helps you understand stability before you commit to manufacturing. Not every lab offering peptide analysis is the right fit. Look for labs with direct experience in your peptide type: linear, cyclic, pegylated, modified, or conjugated. They should be able to troubleshoot, not just produce data. They must operate under GLP or GMP when required, provide transparent documentation, and follow validated analytical methods. The right partner becomes a technical collaborator, not just a vendor. There are several common pitfalls to avoid. Sending samples before understanding stability can lead to sample degradation during shipping. Not defining purity requirements up front leads to rework and delays. Choosing the lowest cost vendor can result in inconsistent or non-regulatory ready data. And treating testing as a final step, rather than an ongoing part of development, can create avoidable setbacks later. Avoiding these mistakes can save months of time and significant cost. A smooth peptide testing workflow follows a logical progression. First, define your specifications clearly. Second, choose testing methods that match your development stage. Third, do early small-scale characterization. Fourth, freeze your specifications before scaling up. And finally, track impurities across every batch for consistency. This creates control, traceability, and regulatory readiness from the start. Peptide testing is not just an analytical requirement, it's a strategic part of development. When you understand what to test, how to test it, and what labs are truly capable, you protect your project from delays and unexpected failures. Define clear specifications early, choose experienced partners, and integrate testing throughout development, not just at the end. This is how you move toward clinical and commercial success with confidence. Thank you for watching.